Good afternoon, students. Today we are going to discuss about the histology of special sensory organs. That is the special organ for the olfaction, that is the smelling, and special sensory organ for the hearing and for vision. These are the three most important special sensory organs today we are going to discuss. First of all, we go for the what is the need of special sensory. This is very, very important to appreciate and to protect and to analyze any stimulus. The special senses are specially designed to perform its functions with the help of nerves, cells, and different structures which are present in the special sensory organs. We see what are the different special sensory structures. What is its microscopic structure? That is nothing but the histology. Histology means, histos means cells. Okay, tissues. What is the microscopic structure of the different sensory organs, especially the special sensory organs we are going to study today class. First, we take about the olfactory special sensory organ. That is, it is the olfaction. It is nothing but the smelling. It is lined by olfactory mucosa. Olfactory mucosa is made up of olfactory epithelium and the underlying connective tissue, that is lamina propria. The olfactory mucosa, it is a special area within the nose. Within the nose, already you know the nasal cavity. First it is a vestibule, then nasal cavity proper. And within the nasal cavity proper, especially the upper or roof of the nasal cavity, especially some part of the medial wall of the nose and some part of the lateral wall of the nose, together they form a specialized olfactory zone. Olfactory zone that is about 2 centimeters in measurement. 2 centimeters in measurement, and that specialized area of the nose, it is responsible for the smell sensation or olfaction. Smell sensation or olfaction. And what are the different structures which are present in the olfactory zone of the nose we are going to study. That is especially the microscopic structure of this olfactory area. Already you know the respiratory, the nose and most part of the nose, it is lined by respiratory epithelium. That is respiratory epithelium, that is nothing but the pseudo-stratified ciliated columnar epithelium. Pseudo-stratified ciliated columnar epithelium, that is in generally the respiratory epithelium. But here the olfaction, it is in the nose itself, but it is having a special epithelium, that is nothing but the olfactory epithelium. Olfactory epithelium, it is different in structure from the respiratory epithelium. The respiratory epithelium depending upon its function. Here, whatever may be the structure in our body, it is modified according to its function. That is the basic point. That is the fundamental point. You take the any part of the body. Suppose if you take the intestines, that is it is having a GIT, gastrointestinal tract, stomach, esophagus, stomach, duodenum, small intestine, again jejunum, ileum, like that. Each and every part of the GIT, it is having its own specialty. At the same time, it is having basic structure and modifications depending upon the function. Depending upon the function of the particular part of the body, there is a modification in the structure. That is especially the microscopic structure of the any part of the body. It is modified according to its function. According to its function. That is the basic fundamental point you have to remember. Here, the olfactory epithelium, it is nothing but the part in the respiratory tract. Okay. And remaining part, it is lined by respiratory epithelium. Only the olfactory zone, olfactory area only, it is lined by olfactory epithelium. And what are the different components of the olfactory epithelium we are going to see. And what are the specialities of the olfactory epithelium. First, if you see the olfactory epithelium, It is a yellow colored zone, about 2 centimeters in measurement, and it is having two parts, especially the lining epithelium, olfactory epithelium, and lamina propria. Below the epithelium, there is a connective tissue part. That part is called lamina propria. Lamina propria. Okay. First, you see the lamina propria contains serous glands, bundles of olfactory nerves, and blood vessels and lymphatics. Okay. These are all the parts which are present in the lamina propria of the olfactory mucosa. Lamina propria of the olfactory mucosa. It contains serous glands. They are also called Bowman's glands, and bundles of olfactory nerves. Bundles of olfactory nerves, blood vessels, and lymphatics. Next, you see here. This is the difference between the olfactory epithelium and the respiratory epithelium. You see here. This is the junction between this part is olfactory epithelium and this part is respiratory epithelium. Respiratory epithelium. Okay, this is the respiratory epithelium up to here, and this is the olfactory epithelium. You see the difference between the the respiratory epithelium. It is modified depending upon its function. What is the function? To eliminate the dust particles, to protect the body and the respiratory tract from the infections, and to eliminate the dust particles. That is the most important. Because of this reason, it is provided with a special arrangement. That is the goblet cells. They are nothing but the unicellular mucus secreting cells, glands, and cilia. That is, they are present cilia. But here there is no cilia. There is no cilia in the olfactory epithelium. That is the most important difference. Here again, this is the respiratory epithelium is pseudo-stratified ciliated columnar epithelium. Pseudo-stratified ciliated columnar epithelium. These are only columnar cells. There may be pseudo-stratification, but there is no cilia, there is no goblet cells. There is no goblet cells. But here it is provided with olfactory nerve endings, that is the receptors, olfactory receptors. Along with that, there is a special arrangement, the serous glands. They are also called Bowman's glands. Bowman's glands are serous glands. Why there is a arrangement of this serous glands? Because the odoriferous substance, first if you want to sense the smell of any substance, first it's supposed to dissolve in the liquid. That is nothing but the serous secretions which are produced by the Bowman's glands or serous glands which are present in the olfactory mucosa. The odoriferous substance, first it dissolves in this serous fluid, then the cells are able to sense the smell. Okay, that is the difference between the olfactory epithelium and the respiratory epithelium. We see in detail what are the different cells which are present in the olfactory epithelium. You see here, the olfactory epithelium is made up of 3 types of cells. There are 3 important types of cells which are present in the olfactory epithelium. The first one is the olfactory cells. That is, they are modified bipolar neuron cells. They are nothing but the modified bipolar nerve cells. They are the olfactory cells. And sustentacular cells. That is nothing but the supporting cells, or tall columnar cells that supports the olfactory nerves. On both sides of these olfactory nerves, there are supporting cells. They are called sustentacular cells. Sustentacular cells. And again, there are another type of cells. There is basal cells, are conical in shape, do not reach the surface and give rise to other cell types. Whenever there is a loss of these cells, there is all sustentacular cells and the olfactory cells. They are replaced by these basal cells. The basal cells, they replace the other types of cells. That is these are the 3 types of cells. They are present in the olfactory epithelium. That is lining epithelium of the olfactory mucosa. It contains 3 types of cells. That is olfactory cells, sustentacular cells, and basal cells. Basal cells. You see here, this is the enlarged micrograph of the olfactory epithelium. Here, these are all the cells, and this is the lamina propria. This is the epithelium, and this is the lamina propria. Here you see in the epithelium, there are 3 types of cells. That is, first one is the sustentacular cells. The sustentacular cells, they are columnar cells, and upper part or apical part is broad and base is narrow. Base is narrow, and here in the upper part, as almost at the level of the middle, they contain nucleus, oval nucleus. Okay, this is the, and in the peripheral, that is apical part, they contain small microvilli. Okay. Next, coming to the another type of cell. This is the olfactory cell. Olfactory cell. This is the bipolar cell, and this is the dendrite. This is the axon. You see here. This is the dendrite. This is the axon. Here, the dendrite of the olfactory cell. It reaches up to the surface of the olfactory mucosa. Then it enlarges and it forms the olfactory vesicle. This is nothing but the olfactory vesicle. This olfactory vesicle, it bears the olfactory hair, olfactory vesicle, and this is the dendrite, and this is the body of the cell, nerve cell, olfactory nerve cell, and this is the axon. It is taking part in the formation of the olfactory nerve, first olfactory bulb and then olfactory tract. Okay, these are the cells, that is the olfactory cells and sustentacular cells. And another type of cell, this is the basal cell. Basal, these are all the basal cell. They are not reaching the top of the mucosa. They are present only at the basal level. And these cells, they replace any loss of the other types of cells. Okay, that is the basal cell. Next, in the lamina propria, these are the Bowman's glands. Bowman's glands are serous glands. Serous glands, they open into the surface of the mucosa, and they pour the secretions into the surface of the mucosa. That is nothing but the serous secretion. This serous secretion is very, very important for the smell sensation, because the odoriferous substance must dissolve in the serous secretions of the Bowman's glands. Then only you are able to feel the smell. Okay, that is the thing. Next, it also contains blood vessels, and these are the olfactory nerves. They are formed in the lamina propria. Okay, these are all the different types of cells which are present in the olfactory epithelium. First, there is a olfactory epithelium and lamina propria. Okay, these are all the different parts of the olfactory epithelium. Next, coming to the another important part, that is the eyeball. Another special sensory structure. Here, within the eyeball, there are some important parts. The histology of some parts you must know, because you see here, this is the first anterior 1/6 or 1/5 of this eyeball. It is formed by a transparent membrane, that is nothing but the cornea. And here, after that, this is the sclera, and here this area is called Sclero-Corneal Junction. Sclero-Corneal Junction. And then another important layer, this is the retina. This is the nervous layer of the eyeball, that is nothing but the retina. And what is the importance of this all these layers, and what is the microscopic structure of the cornea? What is the microscopic structure of the Sclero-Corneal Junction and retina? All these things we discuss now. Okay. Next, first you see about the cornea. Cornea, it is nothing but it is a colorless, transparent anterior 1/5 of the outer coat. The eyeball, it is having three coats: outer coat, middle coat, and inner coat. That is, outer coat, it is made up of cornea and sclera, and middle coat, it is made up of choroid, and inner coat, it is made up of retina. And in the middle coat, extension is nothing but the ciliary body, iris. All these comes under the middle coat. And the inner coat, it is made up of retina. That is the nervous layer. That is the actual layer of the sensation of the light it is received. Okay. Here first you see about the cornea. The cornea, it is a colorless, transparent anterior 1/5 of the outer coat. Outer coat, it is divided into two parts. That is, anterior 1/5 is the cornea, and posterior 4/5 is the sclera. Okay, sclera is opaque. But cornea is transparent. What you have seen, black area of the eye, in our eye, that is nothing but the cornea. Here it is an avascular structure. Because of this reason only it is transparent. Because of the avascularity of the cornea, it is transparent. This is mainly responsible for maintaining its transparency. The avascularity is the mainly responsible for the maintenance of its transparency. And corneal epithelium, it is composed of different parts. What are the different layers which are present in the cornea? Here the refractive index of the cornea is more than the lens. Okay, and here it is transparent. Why it is transparent? All these things we discuss. Okay, first it is a avascular structure. That is the very important for the cornea. Okay, next it is composed of corneal epithelium, that is the surface lining, and below that corneal epithelium there is a membrane that is called Bowman's membrane, or it is also called external limiting membrane. External limiting membrane. Okay. Next is the substantia propria or stroma, that is the substance of the membrane. And next is the Descemet's membrane. Descemet's membrane or posterior limiting membrane. Posterior limiting membrane. And finally endothelium. Finally endothelium. Okay. These are all the 5 layers which are present in the cornea. There is, you see, they compare with the blood vessel. If you take artery also, there are 5 layers. That is the endothelium, the lining epithelium, and that is the inner limiting internal elastic lamina, media, and external elastic lamina, and adventitia. Like that, here also there are different layers. That is the corneal epithelium, that is the superficial layer, and next below that there is a Bowman's membrane or the anterior limiting membrane, and substantia propria or stroma. This is the substance of this layer. And next Descemet's membrane, this is the posterior limiting membrane. And finally there is a endothelium, that is internal lining of the cornea. Internal lining of the cornea. These are the 5 layers which are present in the substance of the cornea. You see here, this is the histology of cornea, microscopic structure of the cornea. And here this is the corneal epithelium. That is, here the corneal epithelium, it is about 5 to 6 layers of polygonal cells. Polygonal cells. It is continuously replaced, and it also contains a basal layer. Basal cell layer. Here this is the basal cell layer. The remaining are 4 to 5 layers. Above that there are 5 to 6 layers of lining epithelium. Below that, this is a small area. This is Bowman's membrane. This is a very tough layer. Very tough layer. It is responsible for the strength of the cornea. Strength of the cornea. And below that, it is having huge part of the substance, that is nothing but the substantia propria or stroma. It is almost 90% of the thickness of the cornea. It is formed with the help of the stroma or substantia propria. From here to here, this is the substantia propria. After substantia propria, there is another layer, that is internal limiting membrane or Descemet's membrane. Descemet's membrane. Descemet's membrane. And below that, this is the another layer, that is the lining corneal epithelium. Internal lining corneal epithelium. These are the 5 layers. And next we see the further magnification. You see here, this is the lining epithelium, that is 5 to 6 layers of polygonal cells. Okay. And this is the basal cell layer. Over that, these are all the different layers. And next, below that, here you see a small light colored area. This is nothing but the Bowman's membrane. Bowman's membrane. It is made up of collagen fibers. Collagen fibers, and they are orderly arranged. Orderly arranged. Orderly arranged. And below that, this is the substantia propria or stroma of the gland. That is the cornea. Stroma of the cornea. It is made up of bundles of collagen fibers, orderly arranged, and they are parallelly arranged with the surface of the cornea. They are arranged along with the parallel surface of the cornea. Okay, parallel with the surface of the cornea. Because of this reason, it is transparent. And at the same time, this stroma also contains keratocytes. 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 Okay, that is the stroma. Next, coming to the inner limiting membrane, that is the here the small, again there is a small membrane. This is the internal limiting or posterior limiting membrane, and this is also made up of collagen fibers. And finally, there is a one single layer of lining epithelium. It is that is endothelium. It is made up of whether the squamous cells or simple columnar cells, simple cuboidal cells. It is made up of cuboidal cells or squamous cells. Squamous cells. These are all the 5 layers which are present in the cornea. Which are present in the cornea, and there is no blood vessels in the cornea, but it is having rich nerve supply. It is having rich nerve supply, but there is no blood supply. And the nutrition to this cornea, it gets its nutrition from the tears, from the anterior part, and from the aqueous humor in the posterior part. That is the anterior chamber of the eyeball. Anterior chamber of the eyeball, it is filled with the aqueous humor. The aqueous humor, it supplies to the nutrition to the cornea from its posterior surface, and from anterior surface, it receives its nutrition from the tears. Okay, and here the lining epithelium of the cornea, the superficial most layer, it is having small microvilli. They are responsible for to maintain the layer of tear over the cornea. Tears over the cornea, that is a small area, small superficial most, it is having small microvilli. With the help of this small projections, it maintains the tears, a layer of tears over the cornea, and it prevents the drying of the cornea. Okay, that is the structure of the cornea. The most important thing is here is it is avascular. It is avascular, and it is transparent. It is transparent, and the thickness of the cornea, it is not uniform. It is not uniform. In the center of the cornea, the thickness is only 0.5 millimeters, and the periphery, it is about 1.1 millimeters. 1.1 millimeters, like this. Okay, in the center, it is thin, and in the periphery, it is thick. Okay, that is in the center, it is 0.7 millimeters, and outside, it is 1.1 millimeters. Okay, that is about the cornea. Because of this avascularity, suppose if you take any burns, acid burns, or acid, some people may pour acids accidentally or incidentally, or due to some other firecrackers. Okay, accidentally bursting of the firecrackers, they may cause damage to the cornea. And if there is a opacity, if there is a burns or scar over the cornea, it produces opacity, and so the person is unable to see the objects. During this time, it will be better to go for the corneal transplantation. Corneal transplantation, it is the most successful transplantation because it is avascular. Whenever there is a vascularity, the blood cells comes into the structure, and they may cause, they may responsible for the tissue rejection. But in case of cornea, there is no such a problem because it is a avascular structure. Because of this reason, any person can donate their eyes. The eye, nothing but they may take the cornea, okay, and they just transplant to the cornea to the people who are already suffering with some opacity, corneal opacities. It is a most successful surgery. Because of this reason, the cornea is very, very important clinical. Okay, next, sclerocorneal junction. Sclerocorneal junction. Posteriorly, the cornea continues as the sclera. Already I had shown. Okay, see here. This is the white. This white one is the cornea, and the blue one is the sclera. The blue one is nothing but the sclera. Here, this part is called sclerocorneal junction. Sclerocorneal junction. The sclerocorneal junction, it is also having its own clinical importance. Because of this reason, we are supposed to know what is the sclerocorneal junction, what are the structures which are present in the sclerocorneal junction. Here, this is marked as a shallow depression externally, and is the place where the transparent cornea becomes continuous with the opaque sclera. Cornea is, anterior part is cornea, that is transparent, and the sclera, it is a opaque. because it is having a different structure, that is collagen fibers. They are not orderly arranged, and it is also vascular. It is also vascular. Because of this reason, the sclera is opaque, but the cornea is transparent. Here, the transparent cornea becomes continuous as a opaque sclera at the sclerocorneal junction. Sclerocorneal junction. You see what is the structure and what is the importance of the sclerocorneal junction. You see here, this is the microscopic structure of the sclerocorneal junction. Okay, here see, this is the cornea, and this is the posteriorly it contains the sclera, and here this is the iris diaphragm, and this is the ciliary body, and here this is the cornea, this is the sclera. At this level, this is the area exactly the sclerocorneal junction. Sclerocorneal junction. Here, already I discussed the cornea, last two layers of cornea. That is the decimus membrane and endothelium. These two are very, very important, because the decimus membrane, it continues in the sclera, and there is here actually here at this level it is about 5 to 6 millimeters in thickness, but when it is reaching to the cornea, microns, 5 to 6 microns, but when it is reaching to the sclera, it becomes 10 to 12 microns, 10 to 12 microns in thickness, and then it divides into small branches, and it forms a specialized structure that is nothing but the venous sinuses. Okay, that is what you call trabeculae. They form the trabecular tissue, and these are trabecular tissue together, they form the venous sinus. Okay, because of this reason, that is layer is very, very important. And here, the sclerocorneal junction, it contains an important circular canal lined by endothelium called the canal of Schem. Canal of Schem, or scleral venous sinus, concerned with the drainage of the aqueous humor. This function is very, very, very important. Aqueous humor drainage is very, very important. The aqueous humor, it is nothing but a fluid that is nothing but the dialysate of the blood. It is just like cerebrospinal fluid. Well, cerebrospinal fluid, it is produced from the body of the that is what you call ciliary body, and by the help of the that is the arterial fluxus, it drains, and it is produced in the posterior part of the anterior chamber. Then it is passed into the anterior chamber through the pupil. Then it is absorbed from the anterior chamber. It is absorbed into the circulation. With the help of the canal of Schem. Canal of Schem, or scleral venous sinus. Okay. Here, this is the very, very important. This function is very, very important. Suppose if there is a block or any obstruction to the canal of Schem, it produces increased intraocular pressure. Normally, the intraocular pressure is about 20 millimeters of mercury. But here, if there is a defect in the drainage of this aqueous humor from the anterior chamber, because of the closure or defect in the canal of Schem or scleral venous sinus, it produces abnormal deposition of the aqueous humor, and it leads to a clinical condition that is called glaucoma. Okay, that is why this is very, very important. This canal is connected externally to the anterior ciliary veins and internal to the anterior chamber through the spaces of fontana or iridocorneal angle. At the iridocorneal angle, the spaces of fontana. This is very, very important. You see here, these are the spaces of fontana, trabeculae. Okay, spaces of trabecular network, the spaces of fontana. They absorb that is the aqueous humor from here. This is the anterior chamber. From here, then it drains into this canal of Schem. Finally, this canal of Schem, it is connected with the anterior ciliary veins. Okay. Usually, the canal of Schem, it won't contain blood. Usually, there is no blood in this canal, because only it is draining. Okay, because the canal of Schem, it is specially specially designed. It is oblique and flat. The structure of the canal of stem is oblique and flat. Because of this obliquity and flatness of this canal of stem, it acts like a valve. It acts like a valve. It prevents the reverse flow of blood from anterior ciliary veins into the anterior chamber. Anterior chamber. This canal of stem, it is very usually, normally there is no blood in the canal of stem. Only the aqueous humor is present. It is drained from anterior chamber into the anterior ciliary veins. But whenever there is a venous engorgement or obstruction to the ciliary veins, venous congestion, the canal of stem and the anterior chamber may, that is the canal of stem and anterior chamber may contain that. Okay, that is the another important clinical aspect. Whenever there is a obstruction to the canal of stem, there is a chances for the venous drainage of the anterior ciliary veins. There is a chances for the accumulation of blood or collection of blood in the canal of stem or scleral venous sinus. That is the most important clinical point. Here, that is at where it is. That is at the sclerocorneal junction. Sclerocorneal junction. That is why the sclerocorneal junction. It is clinically. It is very important, and it is also. You see here another space. That is spaces of Fontan. Faces of Fontan are there nothing but the fourth layer, that is the decimates membrane. It divides into trabecular network like this, and it forms the trabecular spaces. They are nothing but the spaces of Fontana at the iridocorneal angle. All these together they continue as the canal of stem and this scleral venous sinus. Finally, this canal of stem or scleral venous sinus drains into the anterior ciliary vein. Anterior ciliary. Okay, that is the importance. You see here, this is the microscopic structure of the sclerocorneal. Here, this is the cornea and this is the sclera. You see the difference. Here, these fibers are evenly arranged and the fiber is transparent. Here, this is the sclera. It is opaque, and this area is the sclerocorneal junction. And here, this is the canal of stem. Canal of stem. That is the importance of the sclerocorneal junction. Next, coming to the most important part, that is the retina. Retina, histology of retina. The photosensitive optic part of retina is principally composed of two layers. The retina basically are developmental also. The developmental also, retina contains two basic layers. That is, the first one is the pigment epithelium. Next is the nervous layer. Pigment epithelium and nervous layer. Here you see developmentally, the retina, it is developed from the optic cup. Optic cup, the outer layer of the optic cup, it is taking part in the formation of the pigment epithelium, and inner layer, it is taking part in the nervous layer of the retina. Nervous layer of the retina. In between these two, during development, there is a space, that is intraretinal space. Intraretinal space in the fetal life. But once it is fully developed, it disappears. Usually it disappears. And this intraretinal space is clinically, it is very important, because whenever there is a increased intraocular tension, there is chances for the detachment of the nervous layer of the retina from the pigment layer. That is called retinal detachment. Retinal detachment. This is it may leads to blindness. It may leads to blindness. That is why this is intraretinal space. It is nothing but the space between the pigment epithelium and the nervous layer. Pigment epithelium and the nervous layer. And that is intraretinal space. That is the importance of these two layers. Next, first you see about the pigment epithelium. It is formed by a layer of cuboidal pigmented cells. Cuboidal pigmented cells attached firmly to the Bruch's membrane of the choroid. Choroid is nothing but the middle layer, middle coat of the eyeball. Middle external coat is the sclera. Next is the choroid, then retina. Here, here the choroid, it is very closely related with the pigment membrane. That is the membrane of Bruch. Okay. The epithelial cells contain basal nuclei, abundant mitochondria, plenty of melanin granules, and many lysosomes. These are all the structures which are present in the pigment cells of the pigment epithelium. Pigment cells of the pigment epithelium. It contains basal nuclei, abundant mitochondria, plenty of melanin granules, and many lysosomes. The basal surface of the cell shows many infoldings of the plasma membrane, and apical surface shows many microvilli. These microvilli continuously phagocytose the tips of the rods. See here. This is the pigment layer. Pigment layer. This is the nucleus, and this is the microvilli, and these are the small projections. Okay, here this surface, it continuously phagocytose this part of the rods. Okay, and here it also contains the lysosomes and the melanin. The melanin is very important because it is a pigment. Here, after the receiving of the photosense by the receptor cells, that is nothing but the rods and cones, the remaining part of the light must be absorbed. This function is performed by the pigment layer. Otherwise, it may reflect. The reflection of the light, it is prevented with the help of the presence of the pigment layer. Pigment layer of the retina. Pigment layer, it absorbs the melanin, it absorbs the light, and it prevents the reflection of the light. That is the most important function. And the lysosomes, they act like a phagocytic in function. Okay, that is the pigment layer. Next, coming to the nervous layer. It is mainly made up of three types of neurons which are in the visual pathway. There are three types of neurons, that is the receptor cells, that is nothing but the cones and rods, and bipolar neurons, and ganglionic or multipolar neurons. These are the three types of nerve cells which are present in the retina. Here actually the retina, it contains 10 layers. That is a ganglionic cell layer, and below that there are 9 layers. And out of these, that is a layer of rods and cones. It contains rods and cones. And next is the another layer, nerve layer, that is the bipolar neurons. And next is the ganglionic multipolar neurons. And it also contains association neurons and glial cells. Glial cells nothing but the supporting cells. The association neurons is nothing but the connecting neurons between these three types of neurons. Connecting fibers between the connecting neurons between these three types of fibers. That is the rods and cones, bipolar neurons, and ganglionic multipolar neurons. In between these neurons, there is a association neurons. And at the same time, there is a supporting cells, that is nothing but the glial cells. These are all the cells which are present in the nervous layer of the retina. Along with this, there is a external and internal limiting membrane. Okay, outer and inner flexiform. See, what are the different layers? These are all the different layers which are present in the retina. You see, further magnification, you see here, this is the pigment epithelium. Then this is the layer of rods and cones, and this is the outer granular layer, and this is the outer flexiform layer, and inner granular layer, and inner flexiform layer, and layer of ganglionic cells, and the internal limiting membrane, and here there is an external limiting membrane. Okay, you see, this is the drawn picture of retina, that is the histology of retina. You see, first one is this is the lining epithelium, that is the pigment epithelium. Next is the layer of rods and cones. This is the layer of rods and cones. Okay, this is the external limiting membrane. This is nothing but the external limiting membrane. Here the external and internal limiting membrane. Here another limiting membrane, internal limiting membrane. The external internal limiting membrane is nothing but the apical and basal part of the Muller's cells. I show you what is Muller's cell. Okay. Next layer is the outer nuclear layer and the outer flexiform layer. Inner nuclear layer, inner flexiform layer, and this is the layer of nerve cells, multipolar nerve cells, and here this is the layer of nerves, and finally internal limiting membrane. Okay, you see here clearly, you can see this is the pigment cell layer. Then this is the layer of rods and cones, and this is the external limiting membrane. This external limiting, you see here, this total you see, this is nothing but the one cell, Muller's cell. Muller cell. This is the nucleus of the Muller cell, and this is the apical portion of the Muller cell, and this is the basal part of the Muller cell. Here, the apical part of the Muller cell, it takes part in the formation of external limiting membrane, and in basal part, it takes part in the formation of the internal limiting membrane. Here, these are the junctional complexes between the neurons, different neurons. These are the junctional complex, the junctional part of the rods and cones. Okay, this is the external limiting membrane. Next is the outer nuclear layer. Outer nuclear layer and outer flexiform layer. Outer flexiform. Nuclear layer means that is a nerve cell body. And here this is the neurons. They are present in the here. They are from the outer flexiform layer. Next is the inner nuclear layer. That is the nucleus of the Muller cells, nucleus of the horizontal cells, and nucleus of the bipolar cells. Bipolar cells. All these together they form the inner nuclear layer. Inner nuclear layer. And next is the inner flexiform layer. This is also again, it is these axons of the different cell dendrites of the different cells. They are taking part in the formation of the inner flexiform layer. Then next is the ganglionic cell. This is the big multipolar cell. Multipolar cell. It is receiving dendrite axons from the different cells. Amacrine cells and the bipolar cells. They are all making synapses with the multipolar cells. Multipolar cells. And finally, there is a layer of nerve cells. Stratum opticum. Stratum opticum, that is nothing but the nerve cell layer, nerve fiber layer. Nerve fiber layer. Finally, there is a last one is the 10th layer, that is the internal limiting membrane. Internal limiting. These are all the 10 layers which are present in the retina. 10 layers which are present in the retina. Okay. First, you see about the different, what are the specialty or what is the differences between the cones and rods. This is also very, very important in examination point of view. First, you see the structure. This is the membranous discs. Next is the outer segment and modified cilium and inner segment, microchondria, cell body, rod fiber, and finally there is a rod spiracle, right? Okay. Next, you see here, this is the cone. This is the cone. This is the membranous disc. Here, the membranous disc of the cone, it continues with the plasma membrane, but here it is not continuing with the plasma membrane. That is cell membrane. Cell membrane. It is separate from the cell membrane here in the rods, but here it is continuing, and here it is cone shape. Here it is the cylindrical shape. Cylindrical shape. And here this is the modified cilium and inner segment and mitochondria, they are present, and cell body, and this is the nucleus. And here nucleus also you see here, this is the dark nucleus, round nucleus, this is the oval pale nucleus, and this is the cone fiber and cone pedicule. It is forming the pedicule, but it is forming the spirule. Okay, this is the difference between these two. And here the number of the cones, rods, that is 120 millions in each eye. 120 million in each eye. But here only 6 million, that is cones are 6 million distributed around the periphery. The periphery of the retina, it is distributed, but it is more concentrated in the center of the retina, and absent in fovea centralis. In the fovea centralis, the rods are absent, but the cones are more in number, large in number in fovea centralis. Because of this reason, this fovea centralis, it is very, very important for the discriminative vision, color vision. Next, sensitive to light of low intensity, that is a dim light or night vision, but these cones are responsible for the daylight and dark, bright vision. Okay, day vision. And low resolution, so image without clear details. There is no clear details, but here high resolution, image is sharp and clear. Not for the color discrimination. Here, this is for the color discrimination. These are all the differences. It is the next microscopic feature of this rods and cones. Here, outer segment is thin, long, and cylindrical, but outer segment is thick and short and conical. That is about 100 to 120 microns, but here it is 65 to 75 microns. The dark round nucleus is situated deep to the external limiting membrane, but here it is close to the external limiting membrane, but nucleus is pale. And the rod fiber ends at the knob-like spherule, but here it forms the expanded, that is pedicule. And groups of rods are connected with bipolar neurons, but cones fibers end in the pedicule having branched expansion. The membranous discs are independent, but here the membranous discs are connected with the plasma membrane. Look like this. These are all the differences between the rods and cones. Next, another important structure, that is the organ of hearing. Organ of hearing, that is nothing but the organ of Corti. Organ of Corti, it is nothing but for the purpose of hearing sense. Okay, which is the receptor sense organ of hearing. Organ of Corti, that is present in the cochlea. It is present in the cochlea of the internal ear. Cochlea of the internal ear, it contains three parts. Basically, three parts. That is, one is the cochlea. That is nothing but the snail shape. Snail shape. That is why it is called cochlea. Next, semicircular ducts. Semicircular ducts. And there is another part, that is a vestibule. Okay, these are all the three parts. Here, out of these, the cochlea, it contains the hearing sensory organ. That is nothing but the organ of Corti. Organ of Corti. And the hair cells of the organ of Corti that function as mechano-electrical transducers. Okay, this is the very, very important function of the organ of Corti. The mechano-electrical transducers, especially the hair cells which are present in the organ of Corti, they function like a mechano-electrical transducer. Transducers means they change from mechanical to electrical energy. Okay, the basilar membrane supports the organ of Corti. Organ of Corti consists of two groups of cells. That is, the first one is the hair cells, that is inner and outer hair cells. Next is the supporting cells, that is inner, outer pillar cells, inner and outer pharyngeal cells, bordal cells, Hansen cells, and in the center of the organ of Corti, there is a canal, that is tunnel of Corti. Tunnel of Corti. You see here, this is the cochlea. This is the cochlea, the part of the internal ear. This is the cochlea, and here the cochlea, it forms 2 3/4 of the circles. 2 3/4 of the circles. This is 1, 2, 3. Okay, 3/4 of the circles, and here it contains bony labyrinth. This is nothing but the spot is called bony labyrinth, and within the bony labyrinth there is a membranous labyrinth. Membranous labyrinth. The membranous labyrinth of this internal ear, it is divided into three parts. It is divided into three parts. That is, the upper part is called scala vestibuli, middle part is called cochlear duct or scala media, this is the scala tympani. The lower part is called scala tympani. Okay, here you see further magnification. This is the scala vestibuli, the scala media, and scala tympani. Okay, in between the scala vestibuli and scala tympani, there is a specialized zone. This is nothing but the scala tympani. This is called cochlear. That is actually the site for the organ of Corti. Okay, organ of Corti. Here, this scala media and scala tympani, they are separated by a membrane. This is nothing but the basilar membrane. Basilar membrane. This is the basilar membrane. And from the internal part, inner surface of the, especially from the medial part, there is one bony projection. Bony projection into the cochlear duct. And then from this, this divides into two parts, upper and lower part. And again, this part is divided into another middle part, that is nothing but the scala media. Scala media. And here this is the organ of Corti. Organ of Corti. What are the different structures which are present in the organ of Corti? We see. You see, this is the microscopic structure and the lower power of the organ of Corti. Next, you see, this is the higher power. You can see this is nothing but the tectorial membrane, and here this is the internal spiral sulcus. This is the tunnel of Corti. This area is called tunnel of Corti, and these are all the pillar cells, hair cells, and phalangeal cells, Hansen cells. There are different types of cells. You see further magnification. You see here. This is the actual structure, organ of Corti, the higher magnification and different types of cells. Okay. Here, what happens in the organ of Corti? Actually, suppose already you know, that is the vibrations, the sound waves, they are coming, they are reaching the tympanic membrane and they produce vibrations in the tympanic membrane, and with the help of these vibrations inside this tympanic membrane, that is in the middle ear, there are ear ossicles. These ear ossicles also they vibrate, and because of this that is a foot plate of the stapes, it is attached to the internal. That is internally it is attached to the this another tympanic membrane, secondary tympanic membrane, and it produces vibrations in the perilymph. There are two lymphs, that is the endolymph and perilymph. Endolymph, it is nothing but that is this area. That is a cochlear duct. Cochlear duct, it is filled with the scala tympani. Scala media, it is filled with the endolymph, and these two are filled with the perilymph. This perilymph, it is interconnected at the top part of this cochlea, that is the fenestra. Okay, you see here, that is the perilymph and endolymph. Here this organ of Corti, it is having perilymph, it is present in the endolymph, and because of these vibrations in the perilymph and in endolymph, it produces movement in this basilar membrane. And here you see, this is the organ of Corti. The organ of Corti, it is having different types of cells. That is, you see, these are the main cells, that is the hair cells. These are the outer hair cells. These are the inner hair cells. Here, the inner hair cells and outer hair cells are the most important cells which are responsible for the creation, receiving of the sound. That is the thing. And they are also supported by the supporting cells. These are all the supporting cells. This is called pillar cell. This is the inner pillar cell, and here this is the outer pillar cell. These are the pillar cells, and these are the Data cells. There are pharyngeal cells. They support the hair cells, and these are the cells of Hansen's cells. And here there are the again this external. Here this is the internal part. This is the external part, or this is the inner part. This is the outer part. Okay, the inner hair cell is only one. There is only one inner hair cell. Only at the particular membrana, that is one layer. It is not only one cell. It is arranged in the single row. The inner hair cells are arranged like a single row cells, but the outer hair cells are arranged in three rows. Three rows of cells are there, and this inner hair cell, it is having stereocilia. These are the hairs are nothing but the stereocilia. Actually, the inner hair cells are shorter than the outer hair cells. The outer hair cells having stereocilia, they are having in contact with the membrana tectoria or tectorial membrane. It is a gelatinous substance. It is made up of gelatinous substance. It is having contact with the tectorial membrane. But usually the inner hair cell, it is shorter than the outer hair cell. There is no contact of the outer stereocilia with the tectorial membrane in resting condition. Whenever there is a movement of this membrana tectoria, you see here, it is like this. Membrana tectoria is like this. Okay, for the cells. Okay, this is the outer cells, 3 layers, arranged in 3 layers. But inner hair cell, usually it is not connected with the tectoria. Then where there is a movement, then only comes in contact with the membrana, under surface of the membrana tectoria. And what is the mechanism of this hearing? That is very, very important. Here, whenever there is a vibration, sound vibration, it produces waves. Suppose if you see a pond, within the pond, if you throw a stone, what happens? It produces waves like this.
Here also the sound waves, they vibrate the tympanic membrane, then ear ossicles. Finally, they disturb this perilymph and endolymph, and it produces vibrations in the basilar membrane. Basilar membrane. At the same time, it also produces vibrations of this basilar membrane, and there is a movement of this basilar membrane. It leads to movement of the hair cells like this. Hair cells like this. Okay, then the stereocilia, they are coming in contact with this membrana, under surface of the membrana tectoria. They receive nerve impulses. They produce the movements in the stereocilia. It produces a change in the deflection, and it stimulates the hair cells, and it causes release of the neurotransmitters. Neurotransmitters at this area. And finally, the sensations, they are carried through the nerves. That is vestibulocochlear. This is the cochlear nerve. Okay, this is the cochlear nerve, and there are some vestibule also. There is another nerve. This is the nerve fibers. This is all nerve fibers. They carry the sensations from these hair cells. From the hair cells. That is why this organ of Corti, it is very, very important in an examination point of view. You must draw the diagram. Okay, that is that way it functions. Okay, see the microscopic structure in the high power. You see, this is the organ of Corti. This small area is called organ of Corti. This is the tectorial membrane. This is the inner hair cell. Inner hair cell. This is the outer hair cell. These are all the supporting cells. Supporting cells. And this is the basement membrane. Basement membrane. Basement membrane. Okay. These are all the structures which are present in the ear, internal ear, especially the organ of Corti. Okay. Then today we discussed about the different special sensory structures. That is, first is the olfactory epithelium. Okay, olfactory epithelium. That is, it is having two types of structures. That is, lining epithelium and the lamina propria. Okay, and it is different from the respiratory epithelium. Respiratory epithelium, that is very important. And here within the olfactory epithelium, there are three types of cells. That is olfactory cells, sustentacral cells, and basal cells. And next, these are all the different types of cells. Next, coming to the eye. That is, in the eye, cornea is very, very important. It is a colorless, transparent, and it is a avascular structure. The avascularity of the cornea, it is very, very important because it is responsible for the transparency. At the same time, it is favoring for the corneal transplantation because there is no vascularity, there is no tissue rejection after corneal transplantation. Because of this reason, the cornea is very, very important. These are all the different layers which are present in the cornea. That is the lining epithelium, that is 5 to 6 layers of lining corneal epithelium. And below that, there is a tough layer, that is a Bowman's membrane. And next is the substantia propria or stroma. This is the most thickness of this cornea. It is occupied by about up to 90% of the thickness is occupied by this substantia propria. And next is the Descemet's membrane. Descemet's membrane is very, very important because we have the sclerocorneal junction. The sclerocorneal junction, it divides into different layers. Then it takes part in the formation of that is trabeculae. Trabeculae, that is trabeculae. These trabeculae are very, very important to absorb the aqueous humor. And these Descemet's membrane, trabeculae, they are lined by endothelium. They are also lined by, here the thickness of the endothelium at that is the cornea, that is only about 5 to 6 microns. But when it is reaching up to this Descemet's membrane, it is more, and it lines this trabeculae. Okay, that is the importance of the cornea. Next is sclerocorneal junction. The sclerocorneal junction histology is also very, very important because here this transparent cornea becomes opaque sclera. At the same time, it also bears a special area, that is the trabeculae along with the canal of Schem. Canal of Schem. This canal of Schem, it is very, very important to drain the aqueous humor. This is the anterior chamber. It is filled with the aqueous. Here,
This is the area it is producing the aqueous humor. Then it goes to the pupil into the anterior chamber, and it is absorbed into the canal of stem, and it is maintained. If there is any defect or any obstruction to this canal of stem, it produces increased intraocular pressure by accumulation of the aqueous humor. Normal pressure is 20 millimeters of mercury. Whenever there is increase in the intraocular pressure, we call it as glaucoma. That is the most common clinical condition that is responsible, that is due to obstruction to the canal of stem. Okay, next coming to the another area, that is this is the microscope. Next is the retina. What are the different layers of the retina? And out of this, here pigment epithelium, that is the single layer. Then there is a nervous layer. In between the pigment epithelium and nervous layer, there is a space. In the developmentally, because these two, they are developed from the optic cup. The optic cup in external part, it is taking part in the formation of the pigment epithelium, and lower inner part, it takes part in the nervous layer. There is a space that is intraretinal space. Usually disappears after the development of the baby child. There is a fetus. But in adults, sometimes there is a chances for the intraocular pressure, or due to some vascular abnormalities, there is a chances for the accumulation of the blood in between these two layers. And it may lead to, that is a detachment, retinal detachment. That is the importance of this layer. Next, what are the different layers? That is, there are 10 layers in the retina. These are all the 10 layers in the retina. And here, this is the diagrammatic astrology diagram you practice. Okay, 10 layers of the retina. And here, out of these, there is a granular cell layer. This is the layer. It is occupied by blood vessels. The blood vessels here, retina, it is inner, outer 3 to 4 layers. 4 layers, they are receiving blood from the choroid. And inner 6 to 7 layers, they receive blood from this granular cell layer. It is loaded with the blood vessels. The granular cell layer is loaded with the blood vessels. These blood vessels, they supply the remaining layers of the retina by the process of diffusion. Okay, these are all the different layers and this of the retina. Okay, next coming to these are all differences between the rods and cones. And you know better, I think in the physiology already they explained very well about the vision and visual pathway, all these things. Okay. Next, organ of Corti. This organic Corti, the function of the organic Corti is the mechano-electrical transducer. It acts like a mechano-electrical transducer. That is, the sound waves are converted into electrical waves. Then you are able to sense the hearing. Okay, these are all the different structures which are related with this is the actual structure of the organ of Corti. Okay, here this is the microscopic structure of the organ of Corti. Okay, these are all the structures that to study in the special senses.